Okay, this is just the beginning. Let's describe now the composition of the nervous tissue. And even though this is such a complicated tissue that forms the beautiful organs of the nervous system, it consists only on two types of cells, really, neurons and neuroglia. The neurons are the one in charge of transmitting the electrical signal, the electrical potential, the action potential. And the neuroglia, didn't put the name, this in here is the name. The neuroglial cells are supporting cells. They are there to nourish the neurons, making sure the environment around the neurons is stable. It has the proper conditions for the neurons to work. They do not propagate or transmit in any kind of electrical signal. They cannot be excitable like the neurons can be. They also, we will see later that one of their functions, well, they will describe them later. They can wrap neurons in several blah blah blah. We'll go there next video. Now let's concentrate our attention. Let's focus on neurons. And the neurons look something like that. I wanted to start with one of my drawings, but number one, I'm a really bad drawing, and number two, this thing doesn't want to work with me pretty well. But neurons, we can, if you want to draw, and I really want to, if you want to draw a neuron, you can start drawing a neuron like any other cell, like we describe in chapter 1, with the cell membrane and the nucleus right in the middle and some organelles on the periphery. The thing is that this one had some prolongations that are called dendrites. There are these hairs right here. Okay, so let's use this picture to describe it. Now, and this is the basic cell, the basic type, or the most common type, I should say, of neurons in the body. So this part of the cell is called soma, or the body of the cell, this one in here. The entire cell it has a cell membrane like any other cell, but since we're talking about neuron, we're going to say or call it neurolemma. Instead of plasmalemma, we're going to call it neurolemma. Now, inside in the cytoplasm, we are going to find the nucleus like any other cell. We can find also other organelles like a lot of mitochondria. These cells have a lot of mitochondria. We have an extensive network of smooth and rough endoplasmic reticulum. Remember, producing fats, and especially in this one, rough endoplasmic reticulum with ribosomes attached that forms the rough endoplasmic reticulum. These ribosomes can also be free on the cytoplasm. And see that and Golgi apparatus, it also has, of course, Golgi apparatus to secrete the vesicles. But see that I'm drawing most of these organelles in the soma, not in these extensions, because in there is where most of the organelles are. It has the cytoskeleton with neurofibers and neurofibrils and neurofilaments that maintain the shape of the cell. Now, the ribosomes, the free ribosomes, and the ones that are attached to the rough endoplasmic reticulum, when you see them under the microscope, they give the cytoplasmic, or it gives the appearance to the cytoplasm of the neurons to be grainy. And they look like little corpuscles that we called Niesel bodies. Now, these Niesel bodies are only present in the soma or body of the neurons. and they are dark. They give a dark coloration to the cytoplasm, kind of gray. So this is what forms the gray matter that we'll describe later of the nervous tissue. When we see gray matter, we can think, well, in there we're gonna find a lot of these parts of the neuron, this section, a lot of soma or a lot of body, neuronal bodies, because that's where the nasal bodies are. What else? Did I mention everything in there? Okay. Now, coming out from the soma, we're going to see a lot of thin prolongations. These ones, they're called the dendrites. These are dendrites, all of this. And the function of dendrites, or what defines a dendrite, is the direction of the flow of information in here, of the electrical current. So dendrites only can transmit action potentials, or receive or send information from the periphery to the soma, and that's what defines a dendrite. Okay, then we're going to have one single and long prolongation, at least in this type of neuron. How long? Well, imagine if I have a neuron stimulating the skeletal muscle that moves the great toe, is going to be from here, from your brain, and the axon is going to travel all the way down to the your great toe. So these axons can be pretty long, can be pretty short, but in general, one neuron has one axon. And again, what defines an axon is that direction of the information, and the information in here flows just away from the soma of the neuron. So dendrites get information towards the body of the neuron, and axons gets information away from the soma of the neuron. So it's kind of like what we described in the previous video, right? This will be the sensory branch. The soma is the integration center, and the axon is the motor or efferent branch. See, almost the same pattern. Okay, these axons and they sometimes, not always, but they can branch, not as much as the dendrites, but they can branch, and they end as this is the end of another axon of the axon of another neuron, and you can see that in here they end in this knob in here, this dilation. See, we call that end of the axon, we call it, well, it has several names: synaptic terminal, axon terminal. There was another one that I forgot. The thing is that it has a lot of names. It ends just in this dilated portion, which is the synaptic terminal. It's called synaptic terminal because this is the part that is going to get in contact to communicate with another cell. And that another cell can be another neuron, like in this example. It can be a muscle cell, like when we describe the neuromuscular junction in muscle tissue. It can be a gland, and all of these form a synapse. So a synapse is the communication or the site where one neuron communicates with another cell. And remember, the second cell or the other cell can be either another neuron, a muscle cell, or a gland cell. So this is the synapse. I'm gonna put this image in here so we can, I never know how to get rid of those. Just to get rid of the drawings. So this is the enlarged, we just zoom in here. This is the synaptic knob. I'm going to zoom in. We described this already in the muscles when we were describing muscles, but let's describe it one more time in here to remember. So this is the synaptic knob, right? Synaptic, well, it's right here. Where is it? Here. The synaptic knob or the synaptic terminal. And it meets or communicates with another neuron. In this case, it's another neuron, but remember, it can be a gland cell or a muscle cell. So this cell, since is after the synapse, we're going to call it postsynaptic neuron. And this cell, since is before the synapse, we're going to call it synaptic neuron. Okay? And in between the presynaptic neuron and the postsynaptic neuron, there is this space that we call it synaptic cleft. Okay, so those are the three structures. They're not touching each other in this, at least in this type of synapse. So at the end, in the presynaptic neuron, this part of the axon is filled with these vesicles, and these vesicles contains these green balls that are supposed to be the neurotransmitter. And the neurotransmitter is just a chemical substance that can be released into the synaptic cleft to induce a change into the postsynaptic cell, the neuron, the cell, the gland cell, or the muscle cell. Now, at this level also we have a lot of mitochondria because this is expensive. This process in here spends a lot of energy that is provided by the mitochondria. So when the action potential, the electrical signal that was initiated at the beginning of the axon reaches the end, the axon terminal, it depolarizes the membrane, it causes some changes in the membrane, and stimulates the fusion of these vesicles to the neurolemma, to the cell membrane of this neuron at this level. And just by exocytosis, if you can remember that process, by exocytosis, the membrane of this neuron fuses with this and empties all the content, all the neurotransmitter within the synaptic cleft. Now, these neurotransmitters are going to interact with specific receptors that are located on the postsynaptic cleft. In the postsynaptic membrane. Now, every cell can have several types of receptors. Receptors for adrenaline, receptors, maybe the blue is receptor for adrenaline and the red one is receptor for acetylcholine. Okay, and let me grab now, just to make my point. Oh, it didn't leave so much space, but this green one are the receptors that are going to interact specifically with this kind of neurotransmitter. So just the green neurotransmitters, let's say that this is acetylcholine, can interact with the green receptors or the receptors for the acetylcholine. There will be another neurotransmitters, let's say red neurotransmitters, say that this is noradrenaline, can be released also, but these one are going to interact only with the receptors for noradrenaline. What I want to say in here is that this interaction between neurotransmitter and receptor is specific. And this interaction will influence or will modify the cell membrane in the postsynaptic cell and can either stimulate or inhibit that cell. Okay, so it can either stimulate to contract, as we described in neuromuscular junction, or to stop the contraction. Most of the synapses work like this. These are chemical synapses because it involves the use or involves neurotransmitter. Some other synapses are electrical, and we don't have the space in here. We don't have a synaptic cleft in electrical synapses. The pre and the postsynaptic membranes are touching each other, and they're like gap junctions or little tunnels where electrical current just flows by diffusion from one cell to another one. Okay, so that was with the synapse. Now, going back again to another important concept in here. Within the nervous tissue, the cells, the neurons, tend to cluster. And a cluster of, or a group of soma or cell bodies in the central nervous system is called nucleus or nuclei. Now, don't be confused with the nucleus of the cell. Clusters of, and this is important to understand the rest. I know now it's kind of, what is she talking about? But it will make sense. Now, clusters of soma or cell bodies in the peripheral nervous system, we're going to call them ganglia. So ganglia and nucleus are just clusters of soma, one nucleus in the central nervous system, ganglia in the peripheral nervous system. Now, the axons can also cluster and form wires, okay? So a group of axons in the central nervous system, they form tracts. And a group of axons in the peripheral nervous system form nerves, okay? So to have that clear in there. Another thing that is important to say in here is that this, in general, the nervous system, when you see any organ of the nervous system, either the spinal cord or the brain, you're going to see darker areas and lighter areas. And we call that, the darker is the gray matter and the lighter is the white matter. The gray matter consists of clusters of soma, because those are the ones that contain the nasal bodies, the ones that gave the gray color to the gray matter. Okay, so in there we're going to find a lot of soma or cell bodies. We're going to find a lot of dendrites and naked axons. Axons that don't have this blue covering in here. Okay, they're just the yellow thing. Naked axons. We call them unmyelinated axons. We'll understand this in few slides. And the white matter is white because consists of myelinated axons. And myelin is this blue structure that you can see wrapping the axon in there. Those are cells, and that is an insulator, just like cables have. And that insulation is white, it's bright white. So that gives the white color to the white matter. Okay, so in the gray matter, we're just going to find this part. Okay, the soma, the dendrites, and maybe some unmyelinated axons. And in the white matter, we're going to find myelinated axons only. Okay, one more thing that I wanted to talk about neurons is the classification of neurons. Not all of them look like the neuron that I just show you, which is the multipolar neuron. We can classify the neurons either on, we have two classifications, a structural classification based on the way how they look, and the functional classification based on their function. So in the structural classification, we have the multipolar neuron, which has the body with several dendrites, as we described before, and one single axon. Okay, that's the typical multipolar neuron. These neurons are only located in the central nervous system. Okay, so this pink area is central nervous system. Now, we have another type of neuron that we don't have it here, is the bipolar neuron. And bipolar looks something like that. Okay, so we have the soma in the middle and two prolongations on either end of the soma. One acts as an axon, one acts as a dendrite. Which one? Well, just for seeing the neuron, you won't know. You have to see the direction of the information. If this branch is sending information towards the cell body, so this is the dendrite, and if this one is sending the information away from the cell body, this is the axon. That's the only way to know. Now, the third type of neurons, according to these classifications, are unipolar, or best when we call them pseudo-unipolar, because they are almost have one branch, but you will see. Unipolar neurons look like this. This is the body of the neuron. They do have one prolongation, only one coming out from the cell body, but from there it splits in two. And again, one acts like a dendrite, and the other one acts like an axon. I was going to say something. Oh, these neurons, the unipolar and the bipolar neurons, are usually located on the peripheral nervous system, not in the central nervous system. Bipolars are kind of rare in our body, but they can be found in some special senses, like in our olfactory mucosa in the nose, our retina, which is formed one of the layers that detects the light waves. What else? Okay, the other classification consists or classifies a neuron depending on their functions. And we have interneurons, motor neurons, and sensory neurons. And this might be easy for you because we know already, let's start with the sensory. Well, we know the sensory division of the nervous system brings information from a receptor. See, in here we have a receptor towards the central nervous system, brain and spinal cord. So the neurons that perform these functions are sensory neurons, and most of them are unipolar, and they are located in one structure in the peripheral nervous system that is called ganglia. Remember that I told you, and especially this one, dorsal root ganglia. Remember I told you that a cluster of cell bodies in the peripheral nervous system are called ganglia, and a cluster of cell bodies in the central nervous system are called nuclei. Okay, so those are the sensory neurons. Okay, this unipolar neuron is unipolar according to the structural classification, but is also sensory according to the functional classification. Bipolar neurons are also sensory neurons. Motor neurons are always multipolar neurons, always. And remember, the bodies of these neurons are always located in the central nervous system, except in the autonomic nervous system that we'll describe later. These neurons are multipolar, and remember, they're motor, so they are going to send information just in one direction, like you can see in here, from the central nervous system, brain and spinal cord, to an effector, which is a muscle. Okay, and the interneurons are right? So they are located in between the sensory and the motor neurons. So all of them are motor neurons. Multipolar neurons as well. All of them, with no exceptions, are located in the central nervous system. These interneurons make up only, I don't know if I'm exaggerating, but it's close to 98% of all the types of neurons in our body. They're not motor, they're not sensory neurons, they're interneurons. So those are the ones that kind of complicate the study of nervous systems, the ones that connect sensory and motor neurons. the ones that, in order for us to integrate all of these stimulus, uh, interconnect sensory neurons from, they're bringing information from our eyes, from our ears, from our taste, memories, and it creates a big mess in there to send the information to a motor or effector neuron. So multipolar neurons, or yeah, let's say it the other way. I think it's easier. Interneurons and motor neurons are multipolar neurons, and sensory neurons are bipolar and unipolar neurons. Got it? So see you in the next slide. Video. I'm going to describe neuroglia, the second type of neurons. The second type of cells in the nervous tissue. See you. One more thing that I forgot to tell you, the special characteristics of a neuron. First of all, it can live forever, over 100 years, and it can be intact. It doesn't get old. Number two, sadly, they live forever, but they do not divide. After the fetal period, they cannot undergo mitosis. Sad fact. So after they die, they cannot be replaced. And the third characteristic is the highly metabolic rate that these cells possess. So they die within minutes if they don't have the proper amount of glucose and oxygen. Those are the two substances that the neuron eats from. Okay, glucose and oxygen. So now I think I told you everything you need to know now about neurons. See you in the next video.